So I'm really delighted to be able to provide an introduction for our last speaker for the semester today. Dr. Noland is an assistant professor in the McElhenney Skeletal Muscle Physiology Laboratory here at Pennington. Holds degrees in exercise and sport science and in physiology. His research interests in substrate metabolism and health and disease with an emphasis in lipid metabolism and obesity, insulin resistance, and diabetes. I think that covers all the conditions.
Yeah.
So with that, I'm going to turn it over to you.
All right. Thank you very much. So I'm willing to bet that this is probably going to be the least formal lecture that you've had in this series. And part of the reason for that is when I guess when I was at your guys' stage, when I was a postdoc, I was a senior level graduate student, I was kind of at the point where I was tired of being lectured to about things if I wasn't particularly interested in it. So I thought, how could I approach this in order to be more, I guess, more useful? So I thought, well, maybe providing some information on the trials and tribulations that we had while going through this lipid metabolism field. Basically, things that we did by trial and error that I wish people would have told me when I was first starting that would have saved us months or years of work. So that's kind of the approach that I'm taking to this, or just a little bit of background information that I wanted to discuss, but I presume that not everybody in here is particularly interested in, so I'm obviously particularly interested in fatty acid metabolism, lipid metabolism, skeletal muscle, liver, but I'm sure everybody does not have that vested interest in it. But I do think that everybody is going to end up reviewing grants, reviewing papers that have this involved. So also the approach that I took was to provide you guys with information that will help you be able to review grants and papers and make sure that everybody else is using conditions that are useful and good. So, with that, I did send out this book chapter that I had contributed, and hopefully everybody got the email that you didn't necessarily need to read the entire thing, because really I'm just using it as sort of background information to provide a little bit about some necessary aspects of fatty acid metabolism that we'll go through. So I presume that this is probably almost common knowledge for most people, but maybe it's not. So this is just a simple model, basically, of the cellular fatty acid uptake and intracellular lipid trafficking, lipid handling. So when basically fatty acid is presented in the circulation, it's either in the form of long-chain fatty acids bound to a protein called albumin, or generally in lipoproteins like VLDL. With the VLDL, it has to be acted upon by a lipase to liberate these fatty acids, so those fatty acids can then enter through the capillary action and form just basically free fatty acids. And probably 30, 40 years ago, everybody just presumed that these fatty acids basically just passively crossed through the lipid membrane because they're lipid structures themselves. The presumption was that they would just pass through the lipid membrane. But basically 25, 30 years ago, there was discovery of these fatty acid transport proteins like FATP or FAT-CD36, or the fatty acid binding protein at the plasma membrane that help facilitate active transport across the plasma membrane. And well, there's a little bit of controversy. I think, generally speaking, they think anywhere from 50 to 75% of the lipid that enters goes through the FAT-CD36, whereas about 25 to 50% goes through FATP. Once the lipid gets into the cell, so if it goes through FATP, it has a acyl-CoA synthetase activity, so it'll naturally produce a long-chain fatty acyl-CoA, which then is bound to acyl carrier binding protein, ACBP. If it goes through this co-transport mechanism, the long-chain fatty acid will enter unesterified and then be bound by a fatty acid binding protein in the cytosol. So you can see that sort of wherever the transport is, whether it's in the circulation or whether it's in the cell, there's basically a carrier protein that helps traffic these things wherever it needs to go. And within the cell, basically the cell has a choice of whether the lipid ends up going into storage, such as in the triglyceride pool, or whether it ends up being utilized as a fuel source. There's even reasonably compelling evidence that even if it ends up being used as a fuel, there's a few studies at least that suggest that it still has to go through entering into the TG pool to start with. So I'm just going to go into a little bit of how this particular pathway works. So when we're talking about storage, you end up storing things in the triglyceride pool, and I have IMTG for intramuscular here, but that accounts that this basically works for any other tissue that you look at. So in the triglyceride pool, basically triglycerides will end up being inert. But then when you want to mobilize that lipid, it gets acted on by ATGL, which is an adipose tissue triglyceride lipase. But that, don't let the name fool you, ATGL is not just an adipose tissue, it's in all tissues. And ATGL acts primarily on triglycerides, and that'll end up liberating a fatty acid as well as a diglyceride. The diglyceride is preferentially acted on by HSL, which is hormone sensitive lipase, which is the protein that's been known for a very long time. It's regulated by phosphorylation. And HSL will then also liberate a fatty acid and produce a monoacylglyceride, which is then broken down by monoglyceride lipase. So this is basically how stored tissue that's storing triglyceride in it can mobilize that intra-tissue storage in order to use it as fuel, which can then enter the mitochondria, and I'll go into that a little bit later. But I do kind of want to mention that another figure that I mentioned in that article, or the chapter, was this energy cost of triglyceride fatty acid cycling. So again, any type of adrenergic stimulus is going to mobilize these lipids from the intramuscular pools or hepatic pools. And this is just sort of a different way to indicate that you are getting mobilization of triglycerides, formed diglycerides, and monoacylglycerides. And there is an energy cost of doing this. And one of the best models of this is post-exercise. For any of you guys that are interested in exercise research, you know that immediately after exercise, your oxygen consumption rate continues to be very high. And a significant amount of that, and that results in burning extra energy. And a significant portion of that energy cost in the post-exercise period is because you keep mobilizing this lipid. There's still that adrenergic stimulus, but you no longer need the energy, so you don't have to burn that fatty acid. So these triglycerides keep mobilizing fatty acid, but it doesn't need to go to the mitochondria, so it just ends up being reincorporated in the triglyceride pool. And in order to do that, you have to convert these fatty acids to acyl-CoAs, and there's an energy cost to that, which basically is 2 ATP equivalents. And so this triglyceride fatty acid cycling is kind of an interesting model of a futile cycle that just mobilizes fat, restores it. And there are some various conditions where this does contribute to excess energy expenditure. So that's basically sort of just background information. I figure a lot of you knew a lot of that already. So part of I guess, to hit the people that are interested in nutrition, look at, this is a little bit difficult to see, but when we look at general dietary fats, this diagram has it broken down basically into two different types of fats. There's medium chain fats and long chain fats. Medium chain fats constitute anywhere from 8 to 12 carbons. And generally they're found in coconut oil, palm kernel, as well as breast milk actually has quite a bit of medium chain lipid in it, particularly goat milk. And medium chain lipids are thought to be used a little bit easier, and I'll get into that in much more detail here in a little bit. But we don't generally consume a lot of coconut oil or a lot of palm kernel. So there's not really a lot of medium chain lipids in our traditional diet. Most of it ends up being in the form of long chain fats. And this particular model also has it broken down into long chain saturated fats and then unsaturated fats. So the long chain fats are basically 14 to 18 carbons. myristate, palmitate, and stearate. In this model, it says stearate's 18-1, but it's not. It's 18-0. The 0 stands for how many saturated bonds it has. And these are generally in butter, lard, a lot of animal meat. And then there's unsaturated fats. So the monounsaturated fats, the best example of that is oleate. It's 18 carbons, has one unsaturated bond. That's found in olive oil primarily, as well as a lot of other animal fat. And then there's polyunsaturated fats, which most people will refer to as either omega-3 or omega-6. And what's important to know about I'm not going to go into a lot of detail about the omega-3s or omega-6s other than to say that when people talk about the omega-6s, I think it's, or omega-3s and omega-6s, I think it's important to understand that the omega-6 end up driving pro-inflammatory responses, whereas omega-3 fatty acids end up driving more anti-inflammatory responses. And these are also termed as essential fatty acids, and that's simply because while we're able to introduce unsaturated bonds at 9 carbons and above, we can't introduce unsaturated bonds below that. So the unsaturated bonds that are found at the 3rd and the 6th carbons in these omega-6 and omega-3 fatty acids, we have to get those through other sources, so we have to consume them. So talking about dietary absorption of fats, I think it's kind of interesting to point out that, again, we don't end up consuming a lot of medium-chain fats or even short-chain fats, but they're absorbed differently than long-chain fats are. So the long-chain fats are probably what everybody's really taught, and it's described over here in this little corner. When you consume a diet that's high in long-chain fats, the micelle acts on it, it gets absorbed and forms into chylomicrons and then enters into the lymphatic circulation and then gets put back into the circulation after that. But short and medium-chain fatty acids, they don't have the same absorption. They get absorbed basically directly into the portal vein, so they're delivered directly to the liver. And then they can be used as fuel, or they can end up being used for fatty acid synthesis elongation processes. The other thing that's, I guess, of increasing interest now is that since with the explosion in microbiota research, people are particularly interested in the fact that the short-chain fats and medium-chain fats can be fuels for microbiota much more easily than the long-chain fats. Long-chain fats can be modified by microbiota, but I haven't heard of a whole lot of research that says that they're used. So, again, this is just sort of a simplified model here, but it is basically to emphasize the fact that these long-chain triglycerides, they get basically acted upon by micelles and form chylomicrons, and they enter the circulation. They're delivered to peripheral tissues preferentially, whereas the medium-chain lipids are delivered directly to the liver to be processed first. And the other interesting thing is that, again, we don't get a lot of medium-chain lipids in our diet, but studies that have used medium-chain fats or medium-chain triglyceride-enriched diets have noticed that they often actually promote weight loss and can improve insulin sensitivity. And again, part of that is attributed to the fact that they're absorbed in a much different fashion. But the other aspect that really is what people attribute this to is findings showing that they're utilized by mitochondria very differently. So long-chain fats, when we use them, they first have to be converted to an acyl-CoA, which is by an acyl-CoA synthetase. And then they're converted to an acyl carnitine by this enzyme CPT1, carnitine palmitoyltransferase 1. And this is important because neither unesterified fats or acyl-CoAs can enter the mitochondria, they can't cross the membrane. They have to be converted to these acyl carnitines, and then they go through a shuttle system in order to get into the mitochondria to be used as fuel. Alternatively, people have shown that medium-chain fats, they don't require the addition of carnitine or CoA. Medium-chain lipids can enter the mitochondria independent of the CPT1 mediated transport system. So they're much more readily used as fuel because they can enter much easier. And this is the model that's been described for the past 40 or 50 years. But this is where we started. This is where things start to shift a little bit. So when I was in graduate school, we basically were trying to get our system running. So one of the things that we do in our lab is to measure fatty acid oxidation. And to do that, we use radio-labeled fatty acids and measure the release of radioactive CO2. So one of the tests that we wanted to do to make sure our system was working properly is to basically test that model that's been described for the last 40 or 50 years. So we took palmitate and we just left, we had carnitine in the media for some, and then we left carnitine out. So we knew that when we used palmitate, if you don't have carnitine, it can't enter the mitochondria. So that's exactly what we see. We looked at liver, heart, and skeletal muscle, the gastrocnemius. So yeah, you see a 75 to 90% decrease in the oxidation of palmitate when carnitine isn't there. Exactly what we expected. Again, based on the literature that's been, the models been established for the last 40 or 50 years, we expected that when using octanoate, that it wouldn't have an effect because it's supposed to enter the mitochondria independent. That's what we do see with the liver and the heart, but it's obviously not what we see when we look at skeletal muscle. So naturally, since we were just doing this to try to make sure that our model system was working, we thought, okay, maybe we did something wrong with the skeletal muscle, so we tested it a little bit differently. So we took octanoate and octanocarnitine that were radio-labeled, and we did these incubations in the presence and absence of malonyl-CoA, which is a CPT1 inhibitor. And again, what you see in the liver and the heart is that, number one, malonyl-CoA had no effect in either tissue, and adding the octanocarnitine, the oxidation rates were a little bit higher, but maybe 40 to 50% higher. So again, this is just more confirmation that octanoate, at least in the liver and the heart, don't require this carnitine system. Alternatively, when we look at the gastroc, skeletal muscle, we see malonyl-CoA. The CPT1 inhibitor did decrease oxidation 50 to 60%. And then when we used octanocarnitine, it used it about fivefold, at a fivefold greater rate. So again, it looks like carnitine may be really important for octanoate oxidation or medium chain fat oxidation in the skeletal muscle. So we did similar things in isolated mitochondria. So those are in tissue homogenates and have all the cellular components. It's broken up, but generally we thought it would be due to mitochondria. But we needed to test whether it was true in isolated mitochondria. So we took mitochondria from white skeletal muscle and red skeletal muscle, did the incubations in the presence and absence of CoA, carnitine, and then the CPT1 inhibitor. So you can see that when we do palmitate oxidation, when we don't have CoA or carnitine, there's virtually no oxidation, but when CoA and carnitine are present, there's significant palmitate oxidation, and it's inhibitable by malonyl-CoA. So that's, again, just confirming what has been described in the literature for the past 50 years. Alternatively, when we looked at octanoate oxidation, we saw that when CoA and carnitine weren't there, it wasn't oxidized in either red or white muscle very well. Obviously, when adding CoA and carnitine, oxidized very well, and it's also malonyl-CoA inhibitable. So, this is basically just confirming that octanoate oxidation in muscle is very different than it is in other tissues. The other thing that's also kind of interesting to note is that, so when you look at the fiber type comparison, it's pretty common to see rates of octanoate oxidation exceed palmitate oxidation because it's a shorter lipid, it's used a little bit more efficiently as fuel. So when looking at the red muscle, that's basically what you see. The oxidation rates are about 125 nanomoles per mg protein, whereas in palmitate, they're about 80. But that's not true when you look at white muscle. The oxidation rates of palmitate are actually much higher in white muscle than than octanoate. So it seems like maybe the white skeletal muscle has a particular problem using medium chain lipids. So it's another nuance that just hasn't been described in literature. And you know, part of the reason that I wanted to show this is not published work. It's stuff that we need to actually publish, but this is stuff that was developed in my former mentor's lab, and he told me that he didn't want me to write the paper, and that was 10 years ago, and he still hasn't done it. So, it's information that needs to be out there for people that are actually interested in lipid metabolism, differences between different chain lengths. But you know, again, I don't expect you guys to be as interested in lipid metabolism as I am. I think the more salient take-home message from this for you guys is, you know, we went into this thinking that these models have been described for 40, 50 years. This is what we should see. Obviously, we didn't see that. And we tested it in various ways. And we've convinced ourselves that medium chain lipid handling in the skeletal muscle is very different than it is in these other tissues. So, if you get results that sort of contradict what is in the literature, I mean, that's what we're supposed to do. That's what our careers are designed to do. You guys are the next generation of scientists. You guys are going to be writing, and in this case, rewriting the textbooks in the future. So this is, when you get results that are contradictory to what you think, don't just discount it. It could actually be very real and very important. And that's what I think we found here. Obviously, so those studies were all in rat. We wanted to make sure that it applied to human skeletal muscle, so made a tissue homogenate from vastus lateralis and showed that the same thing held true in human muscle as well. When we excluded CoA and carnitine, octanoid oxidation rates decreased substantially, and then also did some stuff. Ron's group, my former mentor's group, did some work with the Ouroboros oxygen consumption system, and showed that indeed when you add carnitine in that type of system in a permeabilized fiber, oxidation rates increase 2 to 3-fold, and that effect was negated when you inhibited CPT1 using malonyl-CoA. We think that at the level of mitochondria and within the tissue itself, this model is very true, but as should come as no surprise every once in a while, unfortunately this metabolic phenotype doesn't seem to be preserved in primary human skeletal muscle cells. Because when we left out carnitine and we add another CPT1 inhibitor at amoxer, we use a long chain fat like oleate, no carnitine, and at amoxer they decrease the oleate oxidation rates of this long chain fat. But when we use octanoate, that didn't happen. This is just another lesson that we learned sort of the hard way. We certainly do, I've done human work, I've done animal work, I've done cell culture work. And you kind of have to learn which model systems you can use to test certain questions. So skeletal muscle cells, especially primaries, a lot of times they do preserve the metabolic phenotype, but it's not always. They're not very good for insulin-stimulated glucose uptake, but you can look at insulin signaling. In this case, obviously the stuff that we see within tissue itself is, like, I don't think we'll be able to test mechanisms using primary cell culture in order to figure out why octanoate handling in muscle is so different. Those are just other lessons that we learned the hard way, that sometimes cell culture is a good model, sometimes it's a very bad model. And you have to figure out, with the questions that you guys are asking, whether it could be a model that you can use for your questions. No, that's just what I see. So, that's kind of the portion on the medium chain fats. I've also mentioned the importance of carnitine in facilitating long chain fatty acid oxidation in all tissues, as well as medium chain fat oxidation within the skeletal muscle. And when I was doing my postdoc, we did a lot of work with carnitine, and we found some interesting things there that I think are worth mentioning as well. So I don't know how many people are familiar with carnitine, but it's considered to be a conditionally essential nutrient. And you can see that the highest content, so it's a conditionally essential nutrient because we do have the capability of producing it, but we also get most of our carnitine sourced through the diet. We get it for omnivorous humans, we get about 75% of our carnitine reserves through the diet. A lot of that comes through red meat, which has high concentrations. There's also pretty good levels in basically just any type of animal meat. And there's also reasonable concentrations in dairy products, but you don't really get much in processed foods or like vegetables. So people that eat vegetarian diets get almost no carnitine through their diet. They end up having to produce it all. So, you know, again, we are capable of synthesizing carnitine, and I'll go into that in a little bit, but multiple tissues do contribute. Liver, kidney, and muscle are the primary tissues that do contribute. Carnitine is also been shown to be very important, not just for the reasons that I mentioned earlier, but for reasons that I'll get into. But in terms of maintaining carnitine status in the body, there's this OCTN2, which is an organic cation transporter, which has high specificity for carnitine. Within the kidney, it is responsible for basically reabsorbing most of the carnitine to make sure that we don't lose it. Probably the best model to stress the importance of OCTN2, this carnitine transporter in the kidney, is this JBS mouse, which is, I don't know if anybody's familiar with it, called JBS because it's a juvenile steatosis mouse. The liver steatosis is so bad that basically these animals were characterized because you can visibly see it through their skin, that their liver is so fatty. And you know, these animals, kind of like suckers and OB/OB/DB/DB type of animals, they're bred selectively based off of a characteristic. And as so often happens when you breed, especially based off of a negative characteristic, what you're actually doing with these animals, you're inbreeding, so you're selecting for a genetic mutation. So in these JBS mouse, they actually have a spontaneous deletion of OCTN2. So they're spilling out all the carnitine in the urine. They get severe steatosis, and they're a very diseased model. And a lot of that can be reversed just by giving them supplemental carnitine. So the other thing, OCTN2 is also very highly expressed in muscle and heart, and this allows these organs to sequester the majority of the carnitine pools in the body. They have 50-fold greater concentration of carnitine in the tissue than there is in the circulation, so they're very greedy with it. Carnitine, like originally, there have been supplementation studies that have been used to treat inborn errors of mitochondrial dysfunction, has antioxidant properties. There's been a lot of research used for cognitive function. But again, what it's most well known for is its role in fatty acid oxidation. So I mentioned carnitine biosynthesis, which is critically important for people that are consuming a vegetarian diet. It's a pretty simple process. There's really only 4 enzymes that are described to be involved. One's within the mitochondria, and the other 3 are cytosolic. I think carnitine biosynthesis is one of the most beautiful examples of integrative, coordinated organ systems physiology, because no one organ system has all 4 enzymes in sufficient quantities to make the carnitine you need. So enzymes in skeletal muscle will end up producing this HTML that ends up being delivered to the liver, which can produce TMABO, and then some gets delivered to the kidney. So, as a physiologist, I think this is just a really interesting example of how organ systems communicate with each other in order to maintain whole body health. And if you read anything about carnitine biosynthesis, most of those reports will say that this first enzyme is the rate-limiting enzyme, which is in the mitochondria. But truthfully, what is rate-limiting is the fact that trimethylysine is the first metabolite that has to be used for carnitine biosynthesis, and it's formed by protein breakdown. So protein turnover is actually the rate-limiting step in carnitine biosynthesis. So we did a study where we gave rats a high-fat diet for 12 months. and then we did carnitine supplementation with them, but I'm not really getting into that. But this is just to emphasize sort of the importance of carnitine biosynthesis. So we saw that with this 12-month high-fat diet, we had decreases in 3 of the 4 enzymes that were involved, so it seems like there's a potential decrease in carnitine biosynthesis. And when we looked at acylcarnitine profiling, we did look at free carnitine levels, and sure enough, we do see decreased free carnitine in skeletal muscle, liver, and the plasma of the animals that were fed a high-fat diet. And this was associated with the impairment in glucose homeostasis and increased HOMA. But it's just kind of an interesting example of being prepared when you're doing your science. As we were analyzing all the groups, we kept seeing one animal that was in this standard chow-fed group that was consistently an outlier. It had very low carnitine levels, very low in the liver and the muscle as well as in the plasma. I mean, it's about 3-fold lower than the average. It was associated with much higher triglycerides and fatty acids, and a HOMA level that was very similar to actually the high-fat diet group. So, and a lot of other parameters were actually weird as well, so we're naturally going to exclude this animal as an outlier anyway. But it just so happened that I was very interested in looking at the carnitine biosynthesis genes, and what we actually found out was that this particular animal had a spontaneous deletion in the 3rd enzyme in this process. And it led to metabolic impairment. So most of the time you're not going to get this lucky that you're going to actually find out why your animal was an outlier. But I think the important take-home message from this is the fact that all of these animal studies, most of these animals are inbred strains. Whenever you have inbred strains, you're going to increase your likelihood of having spontaneous mutations. So don't be surprised if you have an outlier that is consistent sort of across the board. It just so happened that the spontaneous mutation in this particular animal, we happened to identify it because it was important in the process that we were looking into. So just another lesson along the way. But again, within the course of that study, we supplemented animals with carnitine. So as you'd expect, after a year of a high-fat diet, these animals were hugely obese. Most of them were over 1 kilogram, which is very big for a rat. Typically, they're about 250 to 300 grams. So they were about 3 times bigger than the control animals. So they're very severely glucose intolerant. But animals that we gave supplemental carnitine in the drinking water for the last 2 months of that diet had significant improvements in glucose tolerance. Didn't completely normalize it, but I wouldn't expect it to, but the improvements were very significant. We were interested enough in order to do a human trial, and what you see here is that humans that had a normal HOMA, a healthy HOMA value, when we were given carnitine, they didn't improve any, but they didn't really have any room to improve. They were already healthy. The beautiful thing about carnitine supplementation is if you take in more than you need, the kidneys will just get rid of it. You'll just have more expensive urine, essentially. But if your body needs it, it'll be delivered to areas that it needs. So, these individuals that had an impaired HOMA that were glucose intolerant, we didn't recruit glucose intolerant individuals, they just happened to be more glucose intolerant. You can see that they had a significant improvement as well following the carnitine supplementation. Again, it mirrors the rat data where it didn't completely normalize it, but significant improvements. So when we start thinking about the mechanism, we start thinking about what carnitine really does. Again, as I mentioned, carnitine is most widely known for its role in facilitating fat metabolism, fatty acid oxidation. So we measured all those parameters. And there were some interesting differences there, but the most striking difference, this was in the rat study, was the fact that when we gave them the carnitine, so I mentioned that the kidneys will spill out carnitine if it's in excess. the kidneys will also allow you to get rid of excess acylcarnitine, so carnitine that's bound to a lipid. So basically what was happening was these individuals were able to get rid of more carbons in the urine rather than store them in the body. So we started wondering how that happens, and this is another model that I had in that book chapter. You know, again, the most widely known role for carnitine is the fact that it is absolutely required for long-chain fatty acid import into the mitochondrial matrix. But what is much less widely described is the fact that anything that's undergoing beta oxidation and forming these acyl-CoAs, or anything that produces an acetyl-CoA from any substrate, any of these CoAs that are in the mitochondria can actually be converted back to acylcarnitines and readily exported from the tissue. And we looked at cell culture, so this is a model where cell culture actually worked well for us. When we provided cells with increasing amounts of carnitine, you see much more acylcarnitines, and most of that actually ends up in the cell culture media. So this is total acylcarnitine, so it includes short, medium, and long-chain acylcarnitines. The vast majority of that, probably 80% of it, is going to be acetylcarnitine. What I found potentially most interesting was the fact that these long-chain acylcarnitines were very readily exported. 90 to 95% of them actually ended up in the cell culture medium. And again, this is basically taking long-chain acyl-CoAs and getting rid of them. And long-chain acyl-CoAs are things that are described in impaired the insulin signaling cascade. So this is getting rid of potentially more toxic type of lipids by allowing these things to be exported from the tissue. So that's basically an underappreciated role for carnitine. The other thing that's kind of important, at least in terms of designing cell culture studies, when you're doing these things, what I've found is that I've reviewed a lot of papers and grants, and you see so many different conditions used, and it's important to be able to look at the conditions and make sure that you're doing the experiments to address your question. So if you're doing experiments in order to look at fatty acid oxidation, the traditional, actually everybody does it this way, is basically you just end up culturing your cells with, at least with muscle cells, you culture your cells having fetal bovine serum in it to grow them, and then you shift them to differentiation media, which just has horse serum. And serum naturally is going to have some lipid in it, it's going to have carnitine in it, but it doesn't have enough carnitine in order to really drive the functions that it needs to. So what people typically do is, they just grow their cells, and then when they do the actual incubation to measure fatty acid oxidation, they just put in a certain amount of carnitine, maybe usually half to 0.5 to 1 millimolar carnitine acutely. So what you see is that that's not really even maxing out the rates at 100 micromolar, let alone 500 micromolar of long-chain fatty acid. So what we did here was we added 100 micromolar carnitine in the differentiation media, which is on the cells for about 4 days before doing the study. And what you see is just adding 100 micromolar carnitine pre-treatment, even if we don't include carnitine in the actual assay itself, that's actually maximizing out rates. So doing the pre-treatment with carnitine is actually, if you want to measure sort of maximal fatty acid oxidation rates in cells, that's going to be the condition that you want to use by including carnitine in the media pre-treatment. And then, when we did the carnitine pre-treatment, as we increased the amounts of carnitine in the media, we actually started to see sort of it's mostly plateaued, but if anything, there's a bit of a decline. And I think that's probably just because the cells are loaded with carnitine, so they're more readily able to export things when necessary. So, again, this is stuff that we haven't published yet, but if you're wanting to measure fatty acid oxidation, I think we need to be putting carnitine in our differentiation media. But if you're looking at lipid storage, and if you're particularly interested in those pathways, most of the time you don't want to favor oxidation. Most of the lipid isn't going to be stored. And that led me to looking at ceramides. So ceramides, a lot of people are probably fairly familiar with ceramides, so this is just sort of a model of where ceramides can be incorporated. They can be incorporated into sphingomyelin, sphingosine, as well as these galacto and glucosal ceramides, which are involved in more structural lipids or complex lipid species. Ceramides are particularly a hot topic in the insulin sensitivity world because they can interfere with insulin signaling, or at least that's what's described. So when we look at ceramide synthesis, basically it starts out with palmitoyl-CoA being attached to serine by serine palmitoyltransferase. And then goes through a series of steps in order to form ceramide. What's kind of interesting is when you look at the structure of a ceramide, essentially what it is a palmitoyl-CoA attached to serine, and then this last step basically just adds another lipid species to it. So it's almost like a diglyceride in structure, where it's two lipids that are bound by a backbone of some sort. It just happens that the backbone here is a serine. So when you look at ceramides, particularly when people are doing, I guess I can mention that in a second. So again, this step, serine synthetase, adds a second acyl-CoA, second fatty acid moiety to it, and that can have various different chain links, and there's 6 different ceramide synthetases that all have different chain link specificity to add. The reason that's kind of important is when we look at the acylcarnitine profile. So when we look at this profile, you see down here the chain link that is listed. It actually represents that second fatty acid that's on the ceramide. The first one's always going to be a palmitate, but they're able to tell by the weight of what the second lipid species is. And what you see is that the most predominant lipid species that are being added to the second step are very long chain lipids. So very long chain lipids aren't particularly common. They aren't found in our diets very much. If they are in our diets, it's basically wax. They don't go into solution very well, so they end up having to be stored in more complex lipid species, so they're stored in ceramides primarily. And what you see here is this particular study compared normal weight active versus normal weight sedentary versus obese sedentary. And what you see is that, generally speaking, these more common lipid species that are in our diet, like 16O and 18-1, 18-1 has a little bit of an increase in ceramides in the obese condition, but generally only 18-1 isn't going to be stored in ceramides, it's going to be stored in triglyceride pools. But what you see is large accumulation of these very long chain enriched ceramides. And so we did a study in Brandymine. It has a muscle-specific CPT1 deficient mouse. And there you see significant accumulation of C16 and C18 within the ceramides. But the very long chain ceramides didn't change. So the reason that this is important is, again, the previous slide that I showed, the very long chain ceramides were accumulating within skeletal muscle of the obese humans. In this particular case, obviously the obese humans are more metabolically unhealthy. In this case, the CPT1 deficient mice, they're actually more metabolically healthy. And they're basically preventing the accumulation of these ceramides that are enriched in very long chain lipids. And there's also been a couple of recent human studies that indicate that it's these very long chain enriched ceramides that are more tightly associated with metabolic disease than these long chain enriched ceramides. So, and this is a little bit frustrating when you look in the literature because I think while most people actually end up having a mass spec analysis of it, so you get all of these ceramide species, what most commonly people do is just add them all together and say this is just total ceramides. But it does seem that maybe chain length is going to be an important factor moving forward when looking at ceramide levels. So for those of you that know my research program, I have a vested interest in very long chain metabolism because very long chain fats can't be used by mitochondria. I'm interested in peroxisomes, and the only way that a very long chain lipid can be broken down is through the peroxisome. So these treatments that are trying to activate ceramidases to break down ceramides in order to improve insulin sensitivity, I think the only way that that's going to work is by also simultaneously increasing peroxisomal function to break down these very long chain fats, because again, they don't go into solution very well, and each fatty acid can be toxic, but very long chain, the general rule of thumb is the more saturated the fat and the longer the chain length, the more toxic they are to the cell. So if ceramidase is breaking down these very long chain enriched ceramides and liberating a lot of this very long chain fatty acid, it can't stick around very long. It has to be either metabolized or reincorporated into ceramides. That's all I'm really going to say about the peroxisome, which I'm sure is going to make people happy. So, we did, but for this reason, I wanted to do some, again, design some cell culture studies to test my ideas with peroxisomes with ceramides. So, tested palmitate only versus a mixture of palmitate oleate, either a 1-to-1 mixture or even a 2-to-1 mixture of palmitate oleate. And we looked at cytotoxicity. So what you see here is that as we increase the fatty acid concentration, if there's a combination of oleate to palmitate that's not toxic to cells at all, you can go out clear to 1.5 millimolar, which is a lot of lipid, and the cells are just fine. And that's simply because the mixture of oleate palmitate, that ends up being incorporated very quickly into triglycerides, and those end up being basically biologically inert within the cell. However, when you just give palmitate, triglycerides don't really like storing 3 saturated fatty acids. So palmitate by itself ends up being quite cytotoxic. And most often what's used in experiments that are designing ceramide loading conditions, they often use 750 micromolar palmitate, which you can see right here, about 25% of the cells die when using this type of condition. And this actually may be conservative because when we did a similar test later, using 750 micromolar, nearly 60% of the cells died. But when we added FBS, that helped things out a little bit, but we still had about 20% of the cells that were dying. So, again, these are, so again, if you're doing cell culture studies to look at ceramides, you want that lipid to be stored. So most of those studies are also done without carnitine. So all of that palmitate that you're loading, it can't necessarily be utilized very efficiently by the cells, by the mitochondria there. It's not being used as fuel. So it's basically acting as a detergent to the membranes, so it can be really toxic. So this was a study that was done by Diana Obanda, who was in Will Cefalu's lab here. And this is actually just treating with 200 micromolar. So part of the issue that I have with cell culture studies, to look at ceramides, even though we have to do them, is when you use palmitate, it's basically the sole source of lipid. So what we have down here is the treatment lipid. She used a lot of different lipids that she exposed to the cells, and then measured the ceramide lipid from anywhere from C16 to 24. And what you see is that by far and away, the lipid that is enriched the most in virtually all these cases is palmitate, which is almost what you'd expect. But you don't get very much enrichment of these lipids 24 and 24-1 when you use this traditional ceramide lipid loading condition. And so again, I mentioned that everything that we got from tissue says that ceramides that are enriched in very long-chain lipids are the predominant species, and those seem to be what are most tightly associated with insulin resistance and disease. But when you do this traditional palmitate loading of cells, you don't get a representative ceramide profile. So that ends up being problematic. The other thing that's a little bit difficult with ceramide studies in the cells is you can actually buy ceramides with varying chain lengths attached. What people typically do is they purchase a ceramide that has a C2, so it just has an acetyl residue on it, and that they hope that it sort of exchanges for another lipid that's in the cell. C2 ceramides can enter the cells fairly well, but when you go up to like a C16 ceramide or the C24 ceramides, they don't readily enter the cells, and even if they did, it's virtually impossible to do that type of study because those ceramides, they don't go into solution almost at all. In order to get them into a solution, you have to use solvent that is basically incompatible with cell life. So if you put that solvent on your cells, it's going to kill them all. So, doing the cell culture models, we're trying to figure out a way to be able to enrich the cells with the C24 lipid, but it's a challenge. These are obviously lessons that I've learned along the way, and if any of you are interested in doing ceramide work, let all of this be a precautionary tale for you. So then we'll move on to albumin. Reason that I wanted to talk about albumin is because when I went to my postdoc, probably the first year, I'd say half of our lab meetings were talking about all of the different problems that you have with different types of albumin. So, albumin, not to short circuit its importance in various things. Found this in order to emphasize that it has a lot of different functions. It's very important in maintenance of blood pressure, vascular pressure, acid-base balance. It has an antioxidant effect. Can transport drugs, has an anticoagulant effect. It's important for microvascular integrity, but what it's primarily known for is a transport protein. It'll transport ions, bilirubin, various hormones, but again, obviously what I'm most interested in talking about today, at least, is its transport of fatty acid. It's a reasonably large protein, about 67 kilodalton, and basically 1 mole of albumin can bind anywhere from 6 to 7 moles of fat, and most studies are done at about a 4 to 5 molar ratio, and that's important when reviewing papers or designing your studies, because the binding kinetics are kind of similar, they're reminiscent, I guess, to hemoglobin, at least with the binding of oxygen. So with hemoglobin, if it's fully saturated, it'll readily get rid of the oxygen, but when you get down to that last oxygen, it's kind of greedy and holds on to it. Albumin's kind of the same with fat. So if you've got basically a 1-to-1 mole ratio of albumin to fat, it's not going to want to let that fat go. But if you have a 5 mole ratio, then it's going to readily get rid of that fat, but allow your cells to take it up. And binding fats to albumin can be difficult. So I don't know how many of you use albumin in your studies. I'm sure most people are fairly familiar with it, but when you go onto the Sigma website, I think there's 50 or 60 different types of albumin, and each has its own special, they're basically separated by they differ based on their approach that was used to purify them. Generally there's, like if you just take a normal cheap fatty, or BSA, it basically just goes through a chromatograph type of separation. If you end up having a 1% solution of it, there's actually going to be 60 to 75 micromolar fatty acid in it. And it's usually sold as 7.5% BSA, which would be anywhere from 450 to almost 600 micromolar fatty acid, which is a lot of fat. It's also going to have other stuff bound to it. We just happen to test the amount of fat in it because obviously we are very interested in fat exposures, and we need to know that the concentrations of lipid that we're exposing to our cells. So, generally we end up purchasing the fatty acid-free stuff because we're a lipid biology lab. But there's also endotoxin-free, globulin-free, protease-free, things that are separated based on ethanol extractions or heat, chalk, and all of them have different reasons why you use them, and I'm going to talk a little bit about the nuances to some of these things because, again, we spent a year trying to figure out what fat or what BSA to use for our different studies. The other thing that you really need to remember is that it's bovine serum albumin, which obviously comes from cows, and much like humans, cows are going to be variable. So the albumin that you get from cows is going to be variable from batch to batch. So what we ended up, and so there can be significant lot-to-lot variability, and that you have the same problem with any serum that you use in cell culture, whether it be the fetal bovine serum, whether it be horse serum. There's going to be a lot of variability from lot to lot. So, and again, anecdotally, I guess 10% FBS that is used in growth media usually has 250 to 100 micromolar fatty acid. So I mentioned that there's going to be a lot of lipid there. So then, because of this lot-to-lot variability, what we typically do is we order a small batch of it, make sure that it functions properly because it's not uncommon to get a lot that does not work well. And if we prove that it works well, we make sure to write down the lot number, contact the company, and make sure that we order enough in order to last us for a couple of years. Because you want your experiments to be as consistent as possible. And we've had stuff where the serum we've ordered the serum, put it on cells, and the serum itself killed cells. And if you order it, the serum is not cheap. So you can spend a couple $1,000s just to pour it down the drain. So again, life lessons that I want to pass on to you guys. So the various BSAs, I mentioned we end up using the fatty acid-free stuff mostly, and that's pretty obvious why we would. We want to control the amount of lipid that we add. But what I kind of want to mention is the importance of the endotoxin-free. So those of you that know about endotoxins know that they basically drive the inflammatory response. And the most, I guess, a common endotoxin that people use is LPS, lipopolysaccharide, which has an obvious antigen recognition site and outer and inner core, which are all different saccharides, and then a polylipid tail, which basically allows it to insert into a membrane. The reason that I'm bringing this up is because when I think of endotoxin and LPS, we think of activation of inflammatory signaling pathways. So a lot of the studies that you look at that have looked at TLR4 signaling, so TLR4 is a toll-like receptor, which can certainly be activated by TNF, which is an inflammatory stimulus. But there's been a lot of research that has used free fatty acids to activate toll-like receptors and shows that this is a lipid-responsive receptor. The problem is LPS will also be a very potent activator of this particular receptor signaling cascade. And when we did a test of our BSAs, so we tested our fatty acid free from Sigma, fatty acid free from a different company called KSE, and then we purchased a fatty acid endotoxin free BSA from Sigma. What you see is when you just add TNF alpha directly, you get massive activation of the TLR4 inflammatory signaling pathway. And about half of that is achieved just by adding fatty acid-free BSA. So fatty acid-free BSA is going to have a lot of endotoxin on it. So in order to do any of this inflammatory, if you want to do lipid-induced inflammation, you really need to use endotoxin-free BSA, which has complications as well. Fatty acid doesn't bind it as well. It's difficult to work with. It's much more expensive. But again, if you're doing inflammation research, or if you're reviewing papers or grants that are doing lipid-induced inflammation, you need to make sure that those studies are done with endotoxin-free BSA. And then I think this is actually the last thing, the importance of globulin-free BSA. So globulins are there's a lot of different types of globulins that generally fall into 4 different categories: alpha 1, 2, and beta 2, and gamma. They're very large, they're larger than BSA, and they generally function in similar fashion to BSA. They function as carrier proteins and coagulation. But where they differ a little bit is their function in inflammation. So when you think of globulins and BSA, something that you probably should consider is that the fact that a lot of people use BSA to block their Western blots. Well, Western blots work by recognizing immunoglobulins. So if your BSA is not globulin-free, what you get is something like this Western that's courtesy of Chris Stadler, where he used BSA, compared it to fish goo, which I don't really know what is in fish goo, versus milk as a blocking reagent. And what you see here is obviously, I don't think I think a lot of times you can end up using a BSA that isn't specifically globulin free and still get away with it because there's not as much inflammation within the cow at that time. But clearly the lot that he got here, he probably wished he would have had globulin free BSA, because what is happening is the BSA is binding to the blot. It has its own globulins, and the secondary is recognizing all of these random globulins that are on the BSA. So yet another lesson learned the hard way of choosing the right BSA. So again, that's basically just a bunch of different life lessons that we learned along the way that I wish I'd known ahead of time that would have saved me months or years of pain in the lipid metabolism world. I don't know that it's going to apply to everybody, because I doubt that everybody's interested in lipid metabolism the same way I am, but at least it'll help you be able to review papers and grants a little bit better. And if you have any questions or any need for help along the way, if you're doing lipid exposure studies, I'm happy to help. But if you have any questions, feel free to ask them, otherwise we can let you go.
So I have one question which refers to kind of the beginning of your talk. You said they can go either to storage or to oxidation of free fatty acids or your long-chain fatty acids. I know, I think there are a handful of papers that show that, at least in muscle or in the heart, that they go via storage to oxidation.
Yeah.
What's the partitioning between, does every fatty acid go through storage first and then to oxidation, or postprandially, for example, with an exercise, maybe not, but...
It's a really good question. It's very tough. So a lot of that stuff being, a lot of that research is being driven, I think, by Matt Watt and some of the researchers in Australia. And there is pretty good evidence in their studies that a lot of it, probably the majority of it, goes into the triglyceride pool and then gets almost immediately liberated again in order to be used as fuel. Difficulty with any of these studies is, again, that's kind of stuff that I tried to emphasize here, is how the conditions of the experiment are designed in order to drive things. So most of the time people are going to design their experiments in order to drive what they're interested in. So lipid exposure conditions could end up driving things into storage first and then going into metabolism later. But I do think, at least from what I've seen, I think a good percentage of them end up going into the triglyceride pool. I couldn't tell you exactly how much, because I really would prefer to see the in vivo dynamics, and I haven't really looked at those particular studies in order to... I think they've done some of the stable isotope tracer types of things in order to get at some of that. I'm not as familiar with that literature, but that's an excellent question. I think it's something that I need to look into. There's a billion papers that I need to read, but too little time. Sorry. Yeah.
Is carnitine, does it have a threshold in terms of loading, like creatine? Like, do you need to get to a certain physiological level to? Because I know a lot of the early creatine studies, there's a lot of, if you're taking it as a supplement, you're peeing it out, basically.
Right.
But if you were loading it appropriate, if you're under loading conditions, under a short period of time, you're able to saturate the system more efficiently, and then you don't have to keep, adding more to the system. Does carnitine have the same dynamics?
That's a really good question. So with carnitine, carnitine is, it's almost similar to glucose. The body really fights to maintain a certain level of carnitine. So like when you load it, the body ends up excreting a lot of it in the urine. So a lot, like the circulating carnitine levels don't really change much. And even within the tissue, doesn't necessarily change much unless you have a diseased state that has sort of artificially low carnitine, then it really helps. So people have done a lot of, like basically tried to use it as a ergonomic aid, and been at best mild results with it. So I think in terms of the loading, I think it only would apply to diseased populations. And in that case, it happens very quickly. The amount of carnitine that you add ends up getting readily taken up by, at least into the heart and the skeletal muscle, and can end up being bound very quickly to these acyl-CoAs and get rid of it. Generally speaking, so you can buy carnitine at GNC or Walmart, like they have supplements there. And so the good thing about it is, at least most of the reports that I've read, you don't see any negative side effects. The biggest side effect is that you might start smelling like fish. But because of the way that the kidney just ends up eliminating everything. So you don't end up having like a carnitine toxicity or anything. The only contraindicated states is that if you're taking a drug that has sort of a lipid structure, like a lot of antipsychotic medications, carnitine will bind it and get rid of it, so it can decrease some other drug effects. But yeah, it doesn't necessarily have the same type of loading profile that you would have with creatine, where you have to do like a stack, anything like that. So
yeah, I guess what's the biological imperative of having enhanced or holding on to carnitine if we're using so much lipid throughout the day? Is there, like, if you take it exogenously, does it downregulate endogenous production of it? Is it costly to make, I guess is the question. Is it biologically expensive to maintain those levels, and is that why I don't know.
That's a good question as well. So I don't think I've ever seen a study that, so I guess the other way to phrase that would be, by taking the carnitine, are you limiting the capacity of your body to be able to produce carnitine when it needs it? So I guess the way that I've designed that study would be to put people on carnitine, so measure activity, their enzymes, put people on carnitine, and then measure the enzyme activity before, during, and after removal of the carnitine to see if the system sort of downregulates. All I can say is that with our carnitine supplementation studies, the animals that were provided supplemental carnitine, that didn't really change a lot of the carnitine biosynthesis genes that we looked at, but we didn't measure activity, which is probably the more relevant variable. So that's a good question, but I don't know.
When you did that study with the carnitine affecting fatty acid uptake, how did you ensure that your tissue homogenate was carnitine deficient? Or was it just so dilute that you assumed that it was?
So yeah, that's a good question as well. So the tissue homogenate definitely isn't carnitine deficient. It's a 20-fold diluted sample, so it's fairly dilute. The carnitine levels would be somewhat low, but again, what we see in our studies, all we do is, when we make our oxidation buffer, we end up adding like 1 millimolar carnitine typically to it. So we just left that out of the reaction buffer, but there is going to be a little bit of carnitine within the tissue, and that's probably why we still have about 10 to 20% oxidation even with palmitate, because there is some endogenous carnitine still within that homogenate. And even with isolated mitochondria, the mitochondria are going to store a little bit of carnitine in them as well to allow that to cycle. So yeah, that's another part of study design, that basically live with what you have.
So you mentioned that most people who are deficient of carnitine are like vegetarians, but what other type of illness or diseases would cause you to have a low concentration of it?
That's a good question as well. So I do want to mention, I think I may have given the wrong impression. So people that are vegetarians, they don't actually have a carnitine deficiency. They have generally lower carnitine, but it's not significantly lower because the biosynthesis can basically take care of what they need. But there are some primary carnitine deficiency diseases, and a lot of that's just because of the carnitine transporter, the OCTN2 is deficient. The other things that are considered carnitine deficiency diseases would be if you have like a CPT1 deficiency that doesn't allow the carnitine uptake into the mitochondria, that's still considered to be a carnitine deficiency because where the carnitine is needed, you basically can't get it there. But yeah, there's primary carnitine deficiency disorders, which are basically like OCTN2 deficiency, where all of it's just spilled out in the urine. But then there's sort of secondary deficiency, which is really more of an insufficiency, and that's where you basically don't consume enough carnitine or you can't produce enough carnitine in order to match the needs. And that's sort of that's more of a, I don't know if there's really a really well-defined limit that you'd have to meet. So, yeah, sorry.
For your peroxisomal fat oxidation, do you think, or in total, do you increase your fat oxidation by that, or do you just replace shorter fatty acids for oxidizing longer fatty acids?
That's a good question as well. So we've done obviously a lot of studies. Again, my lab basically looks at peroxisome deficiency in skeletal muscle. So with peroxisome deficiency, so fatty acid uptake into the peroxisome doesn't require carnitine. So it can, like in these carnitine insufficient states, peroxisomes can help break down these lipids. The difficulty is export from the carnitine. Export from the peroxisome is generally described to be carnitine sensitive, but it's not necessarily required. So yeah, I think that the peroxisome basically serves as a stress relief valve. When the lipid level gets too high for the mitochondria to handle, that the peroxisome's there to sort of relieve some of that pressure. So it can break some of these excess lipids down. And those acylcarnitines are people will generally describe in literature that they can go to the mitochondria to use, but if they're acylcarnitines, they can do the same thing. It is what happens with the other studies that I showed. They can just be readily exported from the tissue as well. So it'd be a way that you could actually break down some of these very long chain lipids, even to just smaller chains, add a carnitine to them, and get rid of them in the urine. Nobody's really been able to do that type of tracking in order to see what the true metabolic fate of those carbons are that goes through the peroxisome, whether it goes to the mitochondria.
Yeah, because that's always my question for mitochondrial oxidation, because the mitochondria, doesn't care where its fatty acids come from. I'm wondering whether you can actually increase the, how do I say that, like the supply of fatty acids into the mitochondria.
Right. Yeah. So I'll say that Henry Gruengraver, Case Western, he's done a little bit of the back in the mid to late 90s. He did a little bit of stabilized tracer type stuff in liver and heart, and I think had a bit of an issue with some of the results in interpretation, I guess. I guess I just differ a little bit, but the take-home message was that the fate of the carbons ends up being different between those tissues, and that I fully believe. So in the heart, he found that a lot of the carbons actually ended up going into producing malonyl-CoA, which obviously would inhibit fatty acid oxidation within the mitochondria. That also would end up meaning that more lipids would be funneled through the peroxisome as well. So it'd be sort of a self-serving type of pathway. Whereas in the liver, those carbons end up either being used as fuel or they end up being incorporated back into like a triglyceride pool. But again, some of those studies are extremely complicated to read and sometimes a little bit overly technical, I think. But yeah, what the peroxisome really does and its primary biological function, I think that differs between different tissues.
So with that, do you do a cell culture experiment to compare the differences?
Yeah, so I think that ends up being something that.
Cell culture, but a laboratory, I guess.
Yeah, so the beautiful thing about doing that type of thing in cell culture, so if you're talking about stable isotope tracer studies, cell culture can be a good model because you can end up doing a lot of different time points and being able to really measure the kinetics of things. You can do it in vivo, but it's a little bit more challenging, especially with mouse models. But yeah, I think that's something that we're trying to move towards in my lab, is be able to actually do some of these stable isotope kinetic studies. Although we have to pay a pretty penny, I guess, in order to get the specific stable isotopes that we can use for cell culture type studies that would have potentially very long chain lipids that have these stable isotope labels. The other thing is, the problem with very long chain lipids is if they're not soluble, getting them in concentrations that are sufficient in cells is difficult. So, yeah, there are a lot of, with the peroxisome question, we have to go at it. It's tough to attack some of that directly, so we have to go at it sort of indirectly and just use a lot of different ways to get at the question. Yeah, we're interested in how the peroxisome breaks down lipid, but it also produces some of these complex lipid species.
So with the cell culture work that you do you use an established cell line to do that, or do you use primary cell cultures?
We've done a lot of different things. So we use C2C12s a lot, where we overexpress or knock down proteins. We are getting ready to get primary cells from our mouse models. We use primary cells from humans, and either individual humans or even pooled human cells from either obese population or lean population or active population. And I've used L6 cells. So, yeah, we do a lot of different things. Keeps me occupied.
Yeah. Other questions?
All right.
Thank you very much.
Well, we've got a lot of things going. So we, our biggest study, I guess the R1 that I have, is we have a muscle-specific peroxisome deficient mouse model, which we're doing a lot of different tests. So we actually have a peroxisome deficient mouse model, a CPT1 deficient mouse, so that we limit lipid metabolism to the peroxisome as well as the amount conjugate, and then we have double that mouse. We see what effect it has on full body health, as well as substrate oxidation, mitochondrial function. So that's the main project. We also do a lot of exercise physiology studies, exercise science stuff, where we train mice on treadmills and running videos and measure adaptations, do cold exposure stuff. Yes. Yeah. I use postdoc.
Okay,
yes.
I don't have my questions. Thank you. Okay.
Get over if you have questions.
Sure.
Come back.
Cool, thank you. I'll have for you too.
Are you the instructional team?
Pardon?
Are you the instructional team?
For the students,
Right, yeah. This one.
that, we would, that's for the postdocs,
I am.
yeah. Oh, you're taking, but you're not taking it for them. No. So I'm the instructor for the students, taking the graduate students, not postdocs.
Yeah, she put it in there, but she couldn't.
Oh, okay, I'll give it to her, I can give her after this, thank you
Okay.
very much.
I didn't know where her office was.
No, that's, I know where to find her. We usually close out, so thanks very much for that, I appreciate it. Interesting. Yeah,
Yeah.
keeps you here day and night too, weekends.
Yeah, it definitely keeps me occupied. I wish I could have avoided all those problems that I showed you guys.
Well,
That
thank
would-
you for sharing. I think that's useful.
Yes.